Okay, we are going to learn a bit about prokaryotic cells via the bacteria. Okay, bacterium is singular, bacteria is plural. They kind of get used interchangeably. So if we just talked about this is a bacterium, but I just have a habit of saying bacteria. I'm just talking about all bacteria in general for this discussion. Okay, so the first thing that we're just going to hit upon is the fact that bacteria are unicellular, meaning it's only one cell for the entire living organism, and that bacteria are often used as the example of prokaryotic cells. Okay, as we have already discussed, you did reading and then watched a lecture, and then you did the opener. Prokaryotic cells are the cells that are without a nucleus. No nucleus. Okay, so here is our picture of a bacterium. Okay, and we're going to label the parts. We're going to say what the parts are. We're going to be filling this in. I'm going to be using some colored pencils for part of this. I did ask you guys to get colored pencils. So if you have them, great. If you don't, then just use a pencil or a pen. Okay, so the first thing we're going to talk about is bacterial genetics. Of course, in a eukaryotic cell, the DNA is located inside the nucleus. For a bacteria, or for bacteria, the DNA, or the genetic information, is wrapped in its one chromosome, typically circular, and that contains all of the genetic information. I do recommend that you write a little bit small. Okay, so here is the circular chromosome. I realize it doesn't really look like a circle. That's just because it gets folded upon itself and like kind of like a shoelace. If I tied a shoelace and then just threw it on the ground, it wouldn't necessarily look like a pretty circle either. What we need to realize is that this one long chromosome contains all of the instructions that are used to build proteins within the cell. Okay, this chromosome, as we've already said several times, not inside of a nucleus. It is considered free-floating, okay, and the area in which it floats is called the nucleoid region. Okay, what's interesting about the bacteria DNA is that it also has these little accessory pieces called plasmids. What's kind of cool about a plasmid is that it is able to leave the bacteria cell through one of these pili, which has several different functions we're going to talk about in a minute. And the pili is able to interact with the pili of another bacterium and transfer this little bit of extra DNA. The reason why that's really interesting is, when we get sick and we get prescribed antibiotics, what we're trying to fight is an illness or an infection that is caused by some bacteria. Some bacteria are harmful to us. Some bacteria are very, very useful. We have bacteria in our digestive system called gut bacteria, and that makes a really big difference to our health, whether we build up the good bacteria in our gut or if we have the bad bacteria in our gut. And a lot of that depends on our own genetics as well as the kind of foods we eat. So when we get sick from bad bacteria, we'll take an antibiotic, right, to help kill this. Well, I'm sure you've heard, or you've probably have heard, of how some bacteria become resistant to antibiotics. And so the way that happens, the way bacteria becomes resistant to certain antibiotics, is because of this extra piece of chromosome. This extra piece is able to be transferred from one bacteria to another through the pili, okay, and these come in contact with other bacterium, and are able to get transferred through the pili, and then that gets inserted into the long piece of DNA, thereby changing the DNA and becoming resistant. So that's pretty cool that that through this little tiny, almost hair-like looking structure that creates an opening from the inside of the bacterium to another pili of another bacterium, and they can exchange these plasmid pieces. Okay, so in this space right here, we're going to get our art on. Need to get some artistic ability because this looks pretty dark right here. So what I'm going to do is I'm actually going to draw it out, and so I would like you to draw it out as well. So how we're going to create this is we are going to work above this box right here. Let me scoot some stuff out of the way. Okay, so we're going to start by just drawing a long tube looking thing. Okay, and then I'm going to grab a colored pencil. Maybe blue wasn't the best color. It's still kind of dark. Okay, and then I'm going to grab so that is going to represent one of the structures that we're going to talk about in a second. And then I'm going to use some red. Oh, I won't use red because that is broken. I'll use some orange instead. Okay. What we're building right now is just part of this part right here that we're going to label. And then finally, I think I'll use some green. Okay, so what am I trying to illustrate here? Okay, and then I'm going to actually, kind of like the picture does, I'm going to go in here and I'm going to color all this dark green. So it kind of looks like my picture. All right, and so what exactly am I trying to illustrate? So we have different components of the bacterium cell, and so part of that is unique to prokaryotes or particular bacteria is this outside region called the capsule. Okay, the capsule is used to help protect the cell of the bacterium from interactions with other bacteria or other just different areas in the environment. So this green piece right here, or maybe I should write it like this. This right here is the capsule. Yeah, I like that better. Okay, then the orange part is actually our cell wall. Now, cell walls are not just unique. Oh, it would help if you could see it. Cell walls are not unique to bacteria because plant cells also have cell walls. Okay, cell walls are extra protection, helps maintain the shape of the bacterium and prevents dehydration. And then finally, we also have the blue part, which is called the cell membrane.